Have you ever wondered how cells divide? It's a fascinating process, and there are actually two main ways it can happen, through mitosis or meiosis. Mitosis is like the ultimate cloning machine. It's a process that allows for growth, repair, and maintenance of our body cells. Picture this. A cell divides into two, and each of the new cells is an exact copy of the original, carrying the same number of chromosomes. This incredible process begins with what we call the prophase. Here, the chromosomes become condensed, the nuclear envelope breaks apart, and spindle fibers start to form. Then we move on to the metaphase, where chromosomes line up at the metaphase plate, like soldiers standing at attention, and spindle fibers latch onto the centromeres of each chromosome. Next, in the anaphase, the sister chromatids take their leave of each other and are pulled to opposite ends of the cell by the spindle fibers. In the telophase, the chromatids reach the opposite poles, new nuclear envelopes form around them, and the chromosomes start to decondense. Finally comes cytokinesis, where the cell membrane pinches inward, slicing the cytoplasm and organelles into two daughter cells. Now let's turn our attention to meiosis. This process is a little more complex as it involves two rounds of division, known as meiosis I and meiosis II. Meiosis is the mastermind behind genetic diversity occurring only in reproductive cells and leading to the production of eggs and sperm. The first round, or meiosis I, we start with the prophase I. It's similar to the prophase in mitosis, but with an added layer. Homologous chromosomes pair up and swap genetic material, a process known as crossing over. The metaphase I sees these homologous chromosome pairs lining up at the metaphase plate with spindle fibers attaching to each pair. Then, in the anaphase I, these pairs part ways and are pulled to opposite sides of the cell. The telophase I sees chromosomes arriving at opposite poles with nuclear envelopes possibly reforming around them. After this, cytokinesis may occur, forming two haploid daughter cells. But we're not done yet. Meiosis II kicks off with the prophase II, where chromosomes condense again. The metaphase II sees chromosomes lining up at the metaphase plate once more. The anaphase II, sister chromatids separate and are pulled to opposite poles of the cell. Finally, in the telophase II, chromatids arrive at opposite poles, nuclear envelopes reform, and cytokinesis occurs, resulting in 4 haploid daughter cells. Each of these cells has half the number of chromosomes as the original parent cell, a key factor in promoting genetic diversity. So, there you have it, the intricate dance of cell division, a process fundamental to life as we know it. Whether it's mitosis, creating identical copies for growth and repair, or meiosis, mixing things up for genetic diversity, these processes are a testament to the incredible complexity and beauty of biology.